Fluorimetry, DSF, sometimes called the Fluorescence Thermal Shift, is a quick and easy way that you can screen proteins' happiness under different conditions. And so when I'm talking about happiness, I'm talking about kind of the sturdiness of the protein, how much heat it takes to unfold the protein or denature the protein. And the way that it works is that you use a qPCR machine to heat up this protein sample, and as the protein sample unfolds, then you have a dye that is going to bind to the unfolded protein. And the way that it does this is because when your protein unfolds, it exposes the hydrophobic regions, the water excluded regions of the protein that are normally in the center of the protein. You heat them up, then they get exposed, the dye binds, and it fluoresces. And this machine, not only does it heat it up, but it also then takes a picture of the fluorescence. And that is going to then allow you to see at what temperature your protein unfolded. And if your protein was sturdier, it was happier under certain conditions, then it's going to have a higher TMA, so an apparent melting temperature. Whereas if your protein was unhappy, then you would have a low TMA, so a low melting temperature. And this is a quick and easy screen that you can do in order to test a variety of different conditions, things like different salts, different pHs, the presence of a ligand, so a binding partner might help your protein kind of sturdify and then increase the TMA. And because the higher TMA means what?
Stronger bonds in between the protein.
Yeah, and when you heat up proteins, you allow them to wiggle, and when they're wiggling, they're able to break free from those non-covalent interactions, those intermolecular forces. And so the more of those forces there are holding the protein together, the sturdier the protein is, the more heat it takes for them to go and unfold. And we call unfolding of proteins, we say they denature. And so the sturdier the protein is, the more heat it's going to take to unfold. And then we just need a way in which we can detect that the protein has unfolded. And so there are different ways that you can do this, but a lot of them require fancy equipment, such as circular dichroism. The beauty of this FTS or this DSF is that all you need is a qPCR machine. And so these are typically used to measure the concentration of things like cDNAs. So to measure expression of a gene, you make the DNA copy of the messenger RNA, so that's the cDNA, and then you measure the copies get made over time. And we are using it, however, for this fluorescence thermal shift. And so this PCR machine can get hot and cold and hot and cold and hot and cold, but we're just gonna have it go from being cold or like coldish to being really hot. And we're gonna have it do it in this gradient ramp. So it's gonna slowly increase the temperature. And as it increases the temperature, every minute it's gonna take a picture along the ramp. And as it's taking the pictures, what's happening is you're seeing fluorescence. And the fluorescence is the indicator that the protein has unfolded. And you can see that you get kind of this curve. So RFU stands for relative fluorescence units. And that's basically the amount of fluorescence. And as you increase, you start at the baseline, you keep going, you're increasing the temperature, but nothing's happening because your protein is still sturdy. But then you reach a temperature at which your protein starts to unfold, and then you get kind of this sigmoidal shape where the protein unfolds, the dye binds, you see fluorescence. Is that you'll see that the curve dips down, and the curve dips down after it reaches the peak, because what happens is it's thought that like the protein then clumps, and when it clumps, it's kind of hiding the binding sites for the dye. And you can see that under different conditions, the protein might have a higher apparent melting temperature. So the TMA is the temperature at which half of the fluorescence is reached. So basically the TM, the melting temperature, would be the amount at which half the protein has unfolded. But what we're seeing is an indirect indicator of that. We're seeing this dye fluorescence, and so then we get is the TMA, the apparent TM, and that is what we can measure based on this. And so it would be the amount, the heat at which you get like half maximum fluorescence if you're doing it this way. There are also like a curve fitting model that we use, but the same basic idea applies. And so what you do is then you can see if a protein has changed its TM under different conditions, such as different pH, different salt, or the presence of a ligand, so a binding partner. You can actually use this as a technique to see if molecules are binding and how strongly they're binding by doing like different concentrations of different compounds. So people often use it as a screening technique to see what conditions their protein might be happy or unhappy at, as well as to see whether they're binding to certain things. And you can also use it in order to see whether if you make mutations to a protein that actually changes the stability of a protein. So there are a lot of really great things that you can use it for as a screening tool. And so if you're only testing a few different things, then you can do it in like PCR strips. But if you want to screen a bunch of conditions, then people often use like a 96 well plate, or even going up to like 384, various things. And a lot of those things, then you're dealing with robots when you get really high up. But with all these, you kind of want to use a white tube. And the white tube, this opaque tube, is going to make it so that the fluorescence doesn't bleed between the different wells or whatever. But you want to make sure that the camera can actually access and to see the fluorescence, and so then and shine through and stuff. And so then you're going to need an optically clear lid. Or they have like optically clear seals that you put on your plate. Because what's going to happen is that up here, there's going to be a camera that's going to actually take pictures of things. And so this is going to heat it up, and this is going to take its picture as you're heating it up. And as you're heating it up, the protein's going to unfold. When the protein unfolds, the dye binds. When the dye binds, you see fluorescence. The dye that we use is like Cipro orange, and it was like not even a dye that's actually like designed for this, but people found that it's really useful to do this. And so this dye is sold as like a protein stain, like a gel stain, but it's really useful for this. You can export the data, and if you export the RFU, the relative fluorescence units, you get this data that then you can upload into a tool called DSF World. And DSF World is a really great website that's going to then allow you to do kind of custom curve fitting to the curves to get a better idea about the actual apparent TM. But at the end of the day, you can hopefully see whether the conditions that you tested have changed the protein's sturdiness or not. And so the higher the TM, or TMA, the sturdier the protein is, basically the more tightly it's held together. Or it can also, often when you bind to a ligand, it kind of sturdifies things up a little bit. And so, but you could also have some, like sometimes you might get an opposite effect type of thing, but then you can at least see a change if it bound compared to if it didn't bind. And so if you see an increase in the TMA, it's kind of indicating that things are getting tighter and held together more tightly. It's taking more heat to unfold them. Whereas if the TMA goes down, then you're destabilizing the protein. And so if you make a mutation to the protein and you see that the TMA goes down, then it might be an indicator that your protein is not liking that mutation that you made to it. Sometimes people use this as a screening tool for then doing some sort of structural biology work. And so maybe they're trying to find an optimal buffer for your X-ray crystallography or for your cryo-EM or various things like this. Screening can be a quick way to kind of optimize your conditions for those other experiments you want to do. And that is the basics of this technique.